speaker is Dr. Besser from Washington State University. And Dr. Besser has been doing a lot of work, as was referred to earlier by Dr. Hyland and others, in the world of bighorn sheep and the world of disease or pathogen transmissions. And so please welcome Dr. Besser.
I've got this microphone. Can you hear me? Is this working? Sort of working? Turn it up. Maybe I'll move it closer. Is that better? All right. I just wanted to start out with a little bit about my background. I'm a veterinarian, University of Minnesota graduate. Came to WSU to spend a year internship in 1981. Stayed for residency in food animal medicine and surgery. Took a job in the food animal clinics. Worked there for about 5 years. Then I switched to a different department with an appointment combined between research and the Washington Animal Disease Diagnostic Lab. And up until very recently, that's been my job ever since. Most of my work's been on livestock. I started working on bighorn sheep pneumonia about 10 years ago with a summer student, an undergraduate student doing a summer project in my lab. And her project actually turned up this bug that was intriguingly titled Mycoplasma ovi-pneumonia, and we followed up on that work, and it's taken up more and more of my time. In the last year, I've switched over, and I'm pretty much working 100% on bighorn sheep pneumonia for at least a little while. I want to talk briefly today about what my research has done to inform what causes bighorn sheep pneumonia, where does the pathogen come from, and what can we do about it. The last one is going to be very short. You know, from the start, it was clear that these bighorn sheep dying in pneumonia outbreaks had terrible lung infections. And microbiologically, they were really complicated with a lot of different bacteria in different parts of the lung. Different animals in the same outbreak would have different bacteria, very complicated. What the key to understanding this disease was to go earlier in the course of the disease and to see that a constant pattern was an initial infection. The first bacteria that got down into the lungs was this bacterium called Mycoplasma ovi-pneumoniae. And the way it works, which was alluded to in a couple of the talks earlier, is it's thought to impair the normal lung defenses, the defenses that keep the bacteria that all animals inhale with every breath from setting up infections. And ovi-pneumonia fairly specifically blocks that lung defense, and that lets all those individual bacteria that would normally be cleared stay there and set up infections. And that's why it's so diverse. The bacteria that are actually in bighorn sheep pneumonia that result in their death are a pretty close reflection of the bacteria that are in the throat of a normal animal, a normal bighorn sheep. So our publication back in Emerging Infectious Disease, we made the statement that MOV pneumonia plays a primary role, meaning it's the epidemic agent that starts the process, causing epidemic pneumonia of bighorn sheep. And I haven't seen anything since then, displayed a lot of work that would make me think otherwise. So I got a lot of words on this slide, but I thought it would be worth going through some of the lines of data and evidence that support that conclusion that MOV pneumonia is the primary infectious agent. As I mentioned, we learned that MOV is the first bacterium to invade the lung tissues. That alone seems to give it a different position than all the bacteria that come afterwards. We looked at MOV pneumonia in bighorn sheep outbreaks, and so far we have not found a bighorn sheep outbreak that is not highly associated with the presence of MOV pneumonia. And we're well over 40 outbreaks where we've had these microbiological investigations to date. We also, in our region, looked at healthy bighorn sheep herds, which I asked the submitter of the data whether they had observed pneumonia in the last 5 to 10 years. And I wouldn't call them healthy unless they've been looked close enough to detect pneumonia if it occurred. We found that 90% of those 30-plus herds showed no exposure to MOV either by antibody or by PCR. Now, this is problematic. Undoubtedly, some of these herds are misclassified. Some of them are based on stronger data than others. But the pattern is so strong, it's really unlikely to reverse. And this is an odds ratio that's very high. As I mentioned before, we detect MOV consistently in the animals involved in these outbreaks. By genotyping the MOV, or DNA fingerprinting, if you like, we can show that it's actually the same strain of MOV that's spreading from animal to animal. within the infection. That's never been done with any of the other pathogens and on a similar scale as we have for MOV pneumonia. Other pathogens do spread, but much more limited extent. We've also, since we learned about MOV and the agencies have been going out actively determining the status of those herds, we've had a number of situations where we were able to observe the onset of pneumonia within herds that had been sampled before that onset of pneumonia. And what we found is that introduction of a strain of MOV into a herd that had previously tested negative for exposure to MOV, or introduction of a new strain of MOV into a herd that had previously been infected with a different strain, coincides with the onset of pneumonia. And we're up to 13 outbreaks now where we have documented that the timing of the onset of the pneumonia coincides with the introduction of the MOV strain. And the year. And lastly, we published on experimental challenge with MOV alone. And by doing that, we've been able to reproduce sort of the key features of the naturally occurring disease: rapid spread and polymicrobial pneumonia, often fatal. Now, I am focused on MOV. I've been accused of being overly focused on MOV, and I admit to it. But I recognize that there are other theories and other factors, and these are always going to be playing a role. These include some you've heard about already: Pasteurella bacteria and all its various names now, lungworms, environmental stresses, nutritional stresses. And my sense is that all of these factors play a role in at least some pneumonia outbreaks, and some of them might play a role in a lot of them. But if they do, the epidemiologic supporting evidence to document that is not yet available. So I have an open mind about these things, but my focus is on the one that does have that epidemiologic supporting evidence and see how far we can get by following up on that. Where does MOV come from? There are some things about mycoplasma pneumonia and other respiratory mycoplasmas that make this potentially an easier question to answer. MOV is not thought to be able to live in the environment more than a few minutes. So you pretty much have to have a live animal source to be able to transmit the pathogen to a susceptible animal. And as Dr. Hyland mentioned earlier, the current, at a textbook level, understanding is MOV infects only sheep and goat species, or caprinae, if you will. There have been unconfirmed reports from other species, including antelope, including cattle, including moose. I've tried to follow up on these wherever that I've observed these reports. And the only one I've been able to get the samples to test it out was the one from the moose in Alaska that the lab reported positive for MOV. And that was actually a different mycoplasma-like organism, as you might expect. So they're unconfirmed. I'm not ruling out that other species might be detected as carrying MOV. But right now I'm sort of going with the understanding of the last 20 years that probably caprinae are they're the elephant in the room. So if you're looking for sources of MOV, you need a live infected bighorn sheep, or a live infected domestic sheep, or a live infected domestic goat, or a live infected mountain goat, or even a live infected muskox. But some of these will be more relevant than others in the area where the disease occurs. I've done most of my work in collaboration with Francis Kassir, who's the biologist in the Hells Canyon area. And for those of you that aren't familiar with it, this is a large canyon system where Washington, Idaho, and Oregon come together. Was native range for bighorn sheep, but they were extirpated in the early 1900s. And then in conjunction with the efforts that were described earlier, they were reintroduced to many levels of the Hells Canyon area. And these grayish polygons are the individual bighorn sheep populations. I don't know, there's 16 or 18 or something like that on there. I've got 2-letter abbreviations for them. The sheep reintroduced there did really, really well at first, and then they proceeded to get pneumonia outbreaks in various places. And I'd like to just walk you through that history over the last 30 years. So got a couple of pictures of some of the research activities there on mortalities so we can do microbiological sampling. And this is largely going to focus on the MOV strain type. This is the genotype or the DNA sequence type of MOV that we can do now to determine what the source of a particular infection was, whether it was one strain or another. And this is a figure from Francis' paper, which I neglected cite here, sorry about that. So we can just sort of go through this. The first observation, recorded observation of pneumonia in the Hell's Canyon was in Lostine in 1986. It had 65% mortality. And we know about this because animals were necropsied at the Washington Animal Disease Diagnostic Lab. Their lung tissues were still in the blocks in the storage room, so we could go back to them, amplify out the mycoplasma genes, and sequence them and determine the strain type. So I'll call that strain 419, or orange strain if you like. Never saw it again in the last 30 years. Now, there was very little sampling done after that because nobody knew to look for anything at that point. There weren't additional deaths. The history shows that there was pretty terrible lamb recruitment for a few years after that outbreak, following which all of a sudden they started rearing lambs successfully again, and they did fine until the next outbreak happened. The next recorded outbreak was in 1992. Again, this is 15 years before I started working on this problem, but there were dead animals in the model, so I could go back, and it was in the Mountain View population, associated with about a 60% mortality rate, and it had a different strain, mycoplasmal pneumonia strain. I'll call it 402 here. And again, never been seen since in all the sampling that's been done since then. The big outbreak happened in 1995, and it affected 4 populations at the north end of Hell's Canyon. I'm going to color code it one in blue, and we don't know what the mortality rate would have been because a lot of animals were removed from that population while sick in an attempt to treat them in captivity. They all died, but we don't know how many would have died had they been left in place. So now I'm going to start going a little faster. This strain, unlike the others that preceded it, was detected again and again in the form of detection in the lungs of lambs with pneumonia for the most part. So 1998, 1999, 2000. Also in 2000, at the Sheep Mountain population at the southern end of Hell's Canyon, a new strain appeared. It was associated with an outbreak of about 65% death loss. And that one also seemed to persist. And I'll just scroll forward here. 2001, 2002. That strain from Sheep Mountain has jumped all the way halfway up the canyon by then. Probably infected a lot of other animals in between that weren't detected. 2003, 2004. 2005, we're back to only the blue strain. 2006. Now in 2007 is about when we had learned about MOV, and all of a sudden we could take the samples that were being obtained from live animals as well as dead ones and look for it. And so you'll see from here on a lot more populations affected. 2008, 2009. This is the last time we observed this yellow strain, strain 415. 10, 11, 12, 13. In 2013, we found a single animal with pneumonia outside of the main black butte population. Did not see an outbreak associated with that at that time, but the following year it moved into the main black butte population and was associated with 33% mortality of all ages. So that's sort of the time lapse of Hell's Canyon, MOV introduction, and pneumonia. But a couple points I just want to bring out is there were at least 5 MOV introductions into Hell's Canyon during this time. We can't be sure that they were free of MOV before that because nobody was sampling and nobody had records. But 2 of these happened a long time ago and have not been detected since. There was probably minimal sampling in this time point, but there's been a lot of sampling since. So I think they're really gone, those 2 strains. 2 other strains got in there and persisted for 10 years or 20 years. And in particular, this blue 404 strain has been associated with ongoing lamb pneumonia year after year, pretty much throughout this. This new strain that appeared in 2013, 2014 has not been seen since. The local population, there was only 6 or 8 animals left alive in that population, and so the decision was made to move them into captivity into research rather than leading them to possibly spread that red strain further. And it has not been seen further, but the blue strain is still there. So where did these come from? We don't know is the answer. But one place we look quite a lot in is other bighorn sheep or mountain goat populations that are surrounding the area. And we can look at a 20-mile radius or a 50-mile radius or probably even a 100-mile radius. We know that they did not come from those species. So that brings me to the other part of this, which is the domestic sheep research that I've been doing. Domestic sheep do frequently carry Mycoplasma pneumoniae. The USDA study, Sheep 2011, that Dr. Howland mentioned earlier, documented it in about 80+% of the operation sample. That was 450 operations across the country. Recently, we randomly picked 10 of those operations and looked for individual animal prevalence. So we tested 10 individual animals from each of those 30 randomly chosen herds. And we got about 60% of the individual animals positive out of that sample selection. Goats have received much less sampling other than what's coming from Dr. Hyland. I looked at 6 herds in the Moscow-Pullman region, and I found MOV in 5 out of those 6 herds. It was about a third of the animals, but there was only a total of 60 animals sampled there. And Laura Heinz's study that was mentioned earlier, we got 16 herds, all in Washington, but some in eastern Washington and central Washington. And she got a reasonably similar figures, about 60% of the herds and about a third of the animals. It's an interesting discrepancy between this and what Dr. Hyland reported, and I think it might have to do with the management of most of those operations as pack goat operations. One other thing we've learned looking at MOV in domestic sheep especially is that they typically don't have one or maybe a handful of different strains like we see the bighorn sheep. Instead, if it's a sizable flock, we often see dozens of different strains circulating at the same time, which is typical of a reservoir species.
Pardon me, did you say the number of strains?
The number of different strains.
How many would this be?
Well, in a couple of herds where we had enough to do a chow estimator, I think we got figures of 28 strains per operation. So I'd like to look at MOV effects in domestic sheep and goats, and I think its statement still holds true that it typically causes relatively little or mild disease, but there certainly are diseases associated with MOV in domestic small ruminants. And coughing syndrome that Marie Belgian talked about for many years, weaned lambs usually, it can be quite severe, but usually the mortality rate is not high, but that's certainly an MOV-like syndrome. There have also been a few very severe pneumonia outbreaks with 10% mortality or higher. Had a chance to work on one of these in goats on the Big Island of Hawaii, and we could document in that case that pneumonia outbreak was synchronous with the introduction of MOV in the form of some breeding goats brought in Texas. And we had some sampling of the herd from before that. And that particular goat herd with 3,000 head was MOV-free prior to the time they brought in those herds. Now, I have 2 things here I want to mention, but they're preliminary data and the analyses are still in progress, so I don't know if the final results, I left out the specifics. We're going back to the data from that big USDA study, Sheep 2011, where we found about 12% of the flocks were MOV negative, and we're comparing their answers to the disease occurrence questions with the 88% of the operations that were MOV positive. And we found that MOV negative herds are significantly less likely to have reported any pneumonia in their operations, or if pneumonia was observed, MOV negative herds reported fewer pneumonia cases than MOV positive herds. So that's also consistent with MOV playing a role in disease in domestic sheep. I also did a study in 2016 using lambs from the University of Idaho Sheep Center in a collaborative project where we compared the growth rate of a cohort of lambs that had been raised from birth without exposure to MOV with another cohort that had been raised normally and were naturally infected with MOV from their dams. So we followed their growth rates between weaning and slaughter, and we had over a 7% decrease in average daily gain in the MOV infected cohort compared to non-infected cohort. And it was interesting, I looked carefully at their lungs at necropsy. There were very, very little, if any, detectable lesions of pneumonia in them. So I'm not sure what caused that decrease in growth. It could be that we saw a bigger decrease in growth soon after weaning, so it could be that most of the lesions happened then, and that actually healed by the time they got to slaughter. Links between bighorn sheep, bighorn pneumonia, and domestic sheep. A lot of this, ones that I find most convincing, come from reports of what happens when bighorn sheep are commingled with domestic sheep. So there are 14 published reports of bighorn sheep being commingled with domestic sheep. This is prior to 2012. In those studies, 88 out of the 90 bighorn sheep died within 3 months of that contact. Other people have reported what happens when bighorn sheep are mixed with cattle or llamas or horses. No pneumonia outbreaks have been reported in those occasions. There has been occasional individual disease, including bronchopneumonia, fatal bronchopneumonia, but overall, cumulatively, less than 10% death loss resulted from those commingling studies. And then recently, there's been a handful of studies where domestic sheep or domestic goats that were selected because they were MOV negative on the routine diagnostic tests were commingled with bighorn sheep. And again, no epidemic pneumonia has been reported, so it contrasts pretty strongly.
Before you go on those reported publications, how many of those were
how many of those were rangeland? The 14 published reports, how much were those experimental and how many rangeland?
All experimental. I'm not even sure experimental applies to some of them because they weren't done as an experiment. They just put them together and observed pneumonia, but they weren't putting them together to find that out.
Yeah, I think for us too, that's something we always want to talk about and think about the differences in terms of stress and what changes in terms.
I'll come back to that. Hold that thought.
Let's wait until after he's done, and then we can ask questions after that, okay?
So my next slide is MOV transmission on the range. So it's a timely question. We've used this DNA fingerprinting to identify strains, and when you find the same strain in animals that have come into contact, it implies strongly that strain transmitted because the diversity of these within MOV is so great, the chances of finding the same strain by chance alone in two animals after they came into contact is very low. So we have examples under natural range conditions where domestic sheep encountered bighorn sheep, both of them carrying the same strain of MOV, and domestic goats and bighorn sheep, both of them carrying the same strain of MOV. Both of these were occurrences that Dr. Wolf detected in Nevada and provided the samples to do it. I want to clarify, we don't know what direction MOV transmitted here. We got them both, they both carrying it at the same time. It could have come from one or it could have come from the other. In the one case, however, a bighorn sheep pneumonia outbreak followed that observation, and so I suspect that it probably was that domestic sheep in that particular instance. When you try to look at what the source is, you want to try to rule out all the possible sources, not jump on any one, likely one. And bighorn sheep, or infected with MOV, or mountain goat infected with MOV, are real life sources of MOV, and they do spread it. I want to make that very clear, that's what I think. However, they are relatively easy to rule out as sources for a new outbreak. So if I go back to Hell's Canyon, I would say all of the bighorn sheep populations outside of Hell's Canyon within probably a 100-mile radius have been characterized for MOV carriage at that time. And they do not carry any of the strains that it spilled over to cause that ongoing pneumonia in that operation. Domestic sheep and domestic goat operations are also plausible sources if they have MOV-infected animals, and they can also be ruled out by sampling and strain typing. It's a little more difficult because you have to consider the possibility of so many more strains. So usually with the wild ungulates, you can pick out how many strains they have by a relatively limited sampling. Domestic sheep and goats have to sample a lot more. But we've sampled so few domestic sheep or domestic goat operations And around any bighorn sheep operation that has a lot of private lands thereabout, it's actually very difficult to get out there and sample them all, I think. My last slide is what can be done about bighorn sheep pneumonia. And here I'm going back to my food animal training. Otto Radistitz at Western College of Veterinary Medicine famously has 3 principles for management of infectious disease: reduce exposure to the causative agent, increase the non-specific host resistance, and increase the specific host resistance. And these, in fact, I think, they pretty much cover the range of what people have suggested as possible things to do to prevent bighorn sheep pneumonia outbreaks. Minimizing contacts with domestic sheep and goats has certainly been on the table for a long time and continues to be there. I've added a second one here that I think shows some promise, which is reducing MOV prevalence in domestic sheep and domestic goat operations adjacent or nearby or within traveling distance of bighorn sheep operations. We've got a couple of management procedures that seem to be able to separate the MOV positive from the MOV negative and could potentially turn a positive flock negative if someone wanted to put in the time and the effort. And there are efforts in bighorn sheep populations to do something about the bighorn sheep to bighorn sheep transmission, and also, like the blue dots in my slide about Hell's Canyon. Those blue dots persisted for 20 years because they are individual carrier ewes. They recovered from the disease if they even had disease, but they are carrying it and they're transmitting it to the lambs and resulting in lamb pneumonia outbreaks year after year. We've now learned that in many of these infected bighorn sheep populations, it's a relatively small number of the ewes that are these chronic carriers. And right now there are test and cull experiments underway in at least 3 states that I know of, where people have been going in, sampling enough to identify who's the chronic carrier and who's not, and removing selectively the MOV carriers or the typhoid carriers, if you will. And the results, it's too early to say how effective this will be, but the results to date look extremely promising. Increasing non-specific host resistance is the second principle, and this would be the best thing by far. If we could make the bighorn sheep more resilient and not have to do all the other things that are necessary to prevent this disease. But the question is, but how do we do that? And we do not know, I think, right now, any way that we can do this. Some of the factors that are plausibly put out there are nutritional or genetic or stress or even population size and structure. But there are probably examples in all cases of populations, for example, bighorn populations that are belly deep in grass and yet experience a terrible pneumonia outbreak, or are widely outbred and genetically look robust, but experience a terrible pneumonia outbreak. So I don't think we can really point to any of these right now and suggest they do it. And increasing specific host resistance means vaccines. And I agree that vaccines need to be looked at, and I will say right here that I'm very pessimistic of getting a good vaccine for MOV in particular in our lifetimes. Not just my lifetime, I'm old, but even it's a really difficult target for a number of reasons that are too long to go into here. I have a lot of folks to thank and a lot of sources for the relatively small amounts of research money that have been spent pursuing this.
Thank you, Dr. Besser.
I wasn't looking at it.
Oh, okay. Are we on time there? We are just a little bit into our break time. We have a break scheduled for right now. If it would be all right, can people ask you questions individually? I know that there are probably going to be questions out there.
Happy to answer questions.
Can we do that? We are going to break for our break right now. We are going to reconvene promptly at 3:30, and we will start